it is my pleasure to begin this third meeting of our session of the Bioinformatics Club seminars. And today we proceed with the discussion of the genetic variation. We've talking about somatic variation. We've discussed germline genetic variants the previous times, both short variants and copy number variants. You can find the recordings on YouTube if you've missed these meetings. And as I've already mentioned, today we're talking about somatic variation, and we have more people and more experts with us today, and I'm excited to introduce. So first we have Dr. Fotis Psamopoulos from Greece, and members of his group, which I hope he'll be able to introduce. So Fotis, please, if you can say a few words about your group and your expertise, and introduce your lab members.
All right, awesome. Many thanks for the introduction, Yuri, and thank you for a very kind invitation. I'd also like to thank Nicola for making this connection at the very beginning. So I'll start with a very big disclaimer. I think I'm far from an expert on anything related to all of those topics. I dabble a bit, so I think I have some better understanding, just enough to say that this is complex enough that no one ultimately is an expert, which is kind of an oxymoron, but you got it. So I'm leading a group here at CERT, the Center for Research and Technology Hellas in Thessaloniki in Greece. One of the institutes of this center is the Institute of Applied Biosciences, which is my institute. And within this institute, I'm directing the Bioinformatics Laboratory, which comprises of multiple people. One of those is here with us, is Ella Fragouli, a PhD student in the group, who is really working on somatic variant calling and very much in depth into that, making her more of an expert than me on this at this point. So that's kind of the very quick and dirty version. I can provide some more context on the background in case it helps down the road. So my background is in electrical engineering, so I'm computer science by training, but I've been doing bioinformatics for the last 20 years at this point. However, although I have kind of smearing of biology context to understand, kind of better discuss with biologists and wet lab people and medical doctors, ultimately my prior expertise is in computer science, algorithms, optimization, stuff like that. Which is quite fortunate because a lot of the big data and NGS data and high throughput data at this point require a lot of expertise in computer science to make things work. Plus, being in bioinformatics, you're able to sort of act as the intermediary between the people who actually have the biological problem and the really hard coding hardware software experts that will be doing the hard part of coding things into very much detail. So with that weird introduction out of the way, I can go directly into the subject of today, which is about somatic variant calling. At this point,
if you don't mind, well, I'm sorry for interrupting.
Yeah,
I will also comment that we have a few more people with us who I hope that will be able to join our discussion and also contribute to discussing these topics. So just a brief introduction. So we have German Demidov, who was the speaker for the last couple of seminars from the University of Tübingen, and we have also one more new speaker with us, which is Ilya Bizin from Croatia, from the EP Value company. And I hope that again these people will be with us for this discussion and will be able to share their opinions. So if you could say hello to the participants. Ilya, can you hear us? Yeah, if you can, please.
Repeat, please.
We hear you. Yeah, just as a way of checking the connection, I asked you to say hello if you can. Yeah. Thank you. Yeah, that's it. Okay. That's all with the introduction part. Yeah, then Fotis, please, the floor is yours.
Yeah, sure, and great to have you also online. I'm really sorry, I'm horrible at kind of tracking out names and affiliations. So at some point, I'll probably ask again, and I deeply apologize for that from the get-go. Right, so somatic variant calling. So far, everyone sort of has heard about the germline. and all the challenges that come with germline kind of issues. So somatic variant calling is kind of a whole beast by itself for one particular reason. So somatic variant calling, somatic variants are essentially mutations of any type, from SNPs to indels to any kind of different kind of variant, that instead of being part of the germline, they appear in small populations of your cells, and therefore during the sequencing process they appear as lower frequencies, allelic frequency if we go in that direction, compared to the germline. That's kind of the quick and dirty version. Because of this peculiarity, that is, the frequencies are largely not 0, 0.5, and 1, but rather any kind of range between 0 and 1, there is kind of a different set of challenges to figuring out whether they exist or not. And these challenges become even more prominent when you start going into low frequencies. aka below 0.05 or even below 0.1, these become increasingly difficult to identify with high confidence. And also, it doesn't help the fact that if you start looking at different computational pipelines, there is not one widely community accepted pipeline or even tool that everyone says that, okay, you should be using that, period. One of the major arguments that replies that I've given back within this my group here, when they ask about, okay, then what tools should I be using? Most of the time my reply is, okay, what do you want to do? And they really hate that. The main argument is that you need to find a tool that better fits the challenge that you're looking into. For example, if you want to talk about variant calling and somatic variant calling, to be specific, and your frequency is relatively high, like over 0.2, that's generally easy enough to figure out with any kind of the major tools. If you want to look at ones like Cerca, Mutek2, FreeBayes, if you want to go in that direction, etc. However, if you want to start looking into lower frequencies, then it becomes a bit more challenging. And the question is, how can you figure out that what you actually report is valid or not? And this is not even going to the question of what are the steps that get you up until the variant calling step. So you have the FASTQ files. Usually most of people do kind of cleaning, filtering, stuff like that to make sure that their data is of good enough quality. But afterwards, there are a lot of steps that are going beyond. I'm going to mapping and then variant calling. Some of those have a significant impact into how well your variant is going to be called. And this impact increases as you go down in frequency. So that's kind of a quick overview of sort of the, well, how it looks like if you start looking about somatic variant calling. And these are sort of the couple of major challenges that you see here. And which comes back to the question of what can we do about it? Looking around literature, I'm sure you've already kind of identified a few systematic comparison of variant calling tools. There are a few. There's definitely one that was published in Scientific Reports a couple of years ago. Well, possibly more than a couple now that I think of it. And they're popping up along the way here and there. Most of them have a common background. They define or they take kind of a data set that they trust. I'll start with the question of what did it mean that we trust a data set? And then they apply different kind of pipelines, and then they compare what they find in common, what is different, and what is kind of expected in the original data. The problem in that case is how you actually define the data itself. What is true, what is not true? How can you be sure that a particular data set is, well, the variants associated with the data set are in your actual data, the FASTQ files, and not just reported, and the ones that are more prevalent. This is where the effort of Stella is going in, which is primarily focusing on figuring out sort of a ground truth, how to do that, and how to effectively benchmark these pipelines. And with that, I think that was a very good segue to give the floor to Stella, which might also have a few slides to present based on that. And I will set up now.
Thank you very much, Fotis, for the introduction. So let me share my screen. Okay, can you see my screen now?
Yep.
Excellent. So in principle, what we have been trying to do was to being able to somehow identify what is being called by the variants. And for that reason, we turned to synthetic data, which I'm going to take a few minutes to explain what they are and what we try to do with it. So synthetic data are, in principle, when you try to catch the underlying mathematical model and the distributions that so you can replicate the data without mimicking it, because we're dealing with patient data. Of course, we have a lot of privacy issues and concerns. So copying data is a big no-no in this field. So somehow we try to generate data that resemble the real data without being a mirror of it. So in our case, because there are, of course, a lot of types of data in the bioinformatics field, we have been playing around with genomics, synthetic data. So in principle, that means that we, at the end of the day, we end up with data in formats that would be the same out of a sequencer. So these are either FASTQ, and in our case, which we are going to talk about in a little bit, BAM files as well. So a little things about the applications in biosciences of synthetic data is that, of course, we can generate data itself. We can perform augmentation. For example, if we do not have enough cases for a use case, then we can replicate that. We can enhance the number of patients or balance classes, because in many cases there are cases of, for example, cases from a specific ailment, but we do not have control cases. So we try to somehow generate those to balance it. And then, of course, we can use synthetic data to protect sensitive data. And like I said before, there are many types of data, from images to tabular data, clinical data, NGS, etc. And of course, we can utilize this kind of trick to train and validate our machine learning models, enhance the statistical power of our studies, and also help support the development of novel algorithms, which we are going to focus on in a few moments. So what we have been doing is that for the purposes and the needs of our institute and, of course, our team, we have been developing some ways to somehow shed light on somatic variant calling. And we have been very active as a team in the Elixir, the Elixir, and specifically in the machine learning focus group. And we have sent projects to the biohackathon, last year biohackathon. And the poster of the framework that I'm going to present is traveling next month to Uppsala for the Elixir all hands meeting in Sweden. And so, yeah. So what's the motivation behind everything I've been talking about? So as you probably are more aware than myself, is that variant calling plays a very important role in precision medicine pipelines, because it helps us identify genetic lesions and spot variants in the DNA. So, but of course, things get more tricky when we go to the low frequency domain, because we cannot discern that easy which variant caller calls what, and if it is an artifact from the sequencing process, if it is noise, or it's due to the purity of the tumor, etc. So one persistent problem that we keep finding and we keep stumble upon, like Fotis mentioned, is the absence or the scarcity, not complete absence, the scarcity of a ground truth data set where everything is well defined beforehand. So you know what is where and how much of it is there regarding the allele frequency of the variants. So yeah, if you have this kind of data set, it would somehow make your life easier because you would know which mutation is at what point, what chromosomal position, and then you can easily test and benchmark the various variant colors, yes. So for example, I have put here a Venn plot. It was one of the first things that I did when I began my PhD. So I took a training data set for training purposes, a tumor normal data set, and I went it through the various somatic variant colors. And so I made this plot with they're more of a disagreement rather than agreement. And so I showed that to Fotis and he said, cool, now that you have it, what are you going to do with it? Who is telling the truth and who has the false positives? And I was like, I don't know. So somehow we wanted to do this about something. We had to do something about the ground truth, and somehow, given our backgrounds, we thought that synthetic data would be a nice idea. So this is where we found ourselves in. And so I looked around and I found an algorithm that is called NEAT, which seemed cool and was easy to play around with. So what I did was that I presented with a reference FASTA file, specifically for the TP53 gene, because it was it plays an important role in the precision medicine, and also it's quite small. It's like 20,000 base pairs. So it was well defined and small and the region was small. So I said, okay, let's go with it. So what NEAT does is that it creates a BAM file. The BAM file is the alignment in a binary format, but you do not have to realign it. So this was very important for us because mapping is one of the 4 major pillars of variant calling, but there is a source of errors because of the mistakes that the algorithm can make. So we had already aligned reads to the reference, so we were ready to take it away to the variant colors. So because we wanted to emphasize on the small frequencies, around 10% and lower, we somehow wanted to take control of the process of the creation. So in principle, what we did was that we created 10 individual files, which then we later merged into one final file. So we somehow, let's say, diluted the allele frequency. So here I have put a toy example that somehow hope it will make sense. So let's say that we have a variant. This is the red triangle, the yellow triangle. So in the case where we have just one read, this would be, of course, 100% allele frequency, because 1 out of 1 means all of them. But if we take this read and combine it with a group that will end up in 10 reads, this from 100% becomes 10%. So in principle, this is the main way that we constructed our data set. So after we had this final BAM file, we were ready to perform the benchmarking. So we picked 5 variant colors, GADK Mutek2, FreeBase, Vardict, Varscan, and Lofrec. All of them, they were used in tumor-only mode. That means that there was no matched normal. So it was just one sample from the tumor, let's say, and we presented the ground truth BAM file to all of them to see what they would pick. So, okay. On one hand, like I said, we wanted to see how well the algorithms, they were able to report the various variants. Yes, the various variants. But on the other hand, we also wanted to see how the variants behave when presented within a range of feature. For example, it will make sense. So regarding coverage, that is how many reads you have per chromosomal position. We wanted to see the relation between the ability of the algorithm compared to the value. So what I did was that I created a series of datasets from 300 coverage all the way to 5,000. And everything else was all the other parameters were at the same value. So this is called an ablation study when you focus on just one parameter. So we presented each algorithm with all this variation of the coverage. And then we repeat that for the read length from just 25 all the way to 300. And of course, we also played with a number of individual files. So in the case of this toy example, instead of having 10 individual files, we had 2 and 4. So I wanted to see if we would have how the allele frequency would be affected, and at the end, the variant colors themselves. So we gave all these data sets, we fed all the data sets to the algorithms one by one and compared all 5 of them towards each of the data sets. And here you see the data set of 1,000 coverage, and the read length is of 150 length. So here we have the box plots of the coverage, the allele frequency, and the allele frequency deviation density plots. So one by one. The box plot for the coverage shows the distribution of the true positive variants. That means that the variants that were present in the ground truth and were able to, and were also detected by its algorithm. So its point is a true positive variant. So here we see that we have some tendencies. Now, one of the algorithms does not take into account all the available information. And this is shown because instead of taking advantage of all the 1,000 coverage, it takes about 1/3 of it, so around 300. So, of course, this, again, we have the allele frequency. This affects the allele frequency as well. And the density plot is translated as we wanted to see not only what variants were detected, but also how much. For example, if a variant was present in the ground truth at, let's say, 30%, we want to check if it was able to be detected by the algorithm, if how much off or not off it was. For example, if it was detected at 25 allele frequency, then we would say that we had 5% off. So this is in principle what you see in the bottom panel. We see that all of the densities, they center around 0, which is good. That means that they are all equal to the ground truth. But again, we have some peaks and some deviations throughout the whole spectrum of the deviation. So our future directions and ambitions is to explore more inherent characteristics of the NGS data maybe add more tumor-only somatic variant calling, and as well explore the tumor normal mode as well. We want to make the pipeline a little bit more user-friendly, so a workflow system like Nextflow is something that we are flirting with at the moment. And also, at this moment, we treat its chromosomal position identically. So we see things very technically without the biological meaning. For example, if it is an exon or an intron, we do not take this into account yet because we focus just on how the algorithms of the variant calling process behave. But in the future, we want to see to extend, for example, the loci that we use, like a whole chromosome, a whole genome, a whole exome, and see how these affect the process as well. Of course, I have not been alone in this journey. I had some amazing supervision and also a lot of help by my teammates from our team and outside of our team. And yeah, this work that I have been describing has been submitted as a preprint in the bioRxiv repository. So if you want to check it out, you can visit that. And yeah, thank you so much for your attention. And I hope everything made sense.
Thanks, Stella.
Yeah, thank you very much for the introduction and for talking about this this method. Yeah, it was very nice. And I think that that's a good point, a good starting point for us to begin the discussion. Yeah, because what you've just presented, basically, yeah, it was a story about checking which of the methods, which of the variant callers produces more or less reliable results. And this is basically what we've been talking about in the last couple of meetings when we were talking about germline variant calling for both short variants and copy number variants. I think we've been we've spent quite a lot of time talking about like the selection of tools and what are the criteria for it. Yeah, so and I think that that's basically could be the starting point for us. So as far as I understand, yeah, given what you were talking about, you have yeah, you see like the benchmarking as the source of like choosing which tool to apply. Is that correct? So basically, I maybe like elaborate on my question, because in the germline field, as we've discussed previously, there is a lot of talks about it. In certain cases, it's better to prefer tools which are maybe better and more widely acknowledged and widely used to ensure compatibility with other studies, and basically to be sure in what are the results. Maybe benchmarks are not like disregarded, but some people in some cases may have like include other factors into the account. So what's your perspective on that? So are you trying to develop this benchmark in order to basically just select a particular tool, or do you have any like preference? Maybe you can comment on the choice of tools which you make in practical applications.
Shall I start and then I give the floor to Stella? The short answer is that we need to figure out what we don't know. The main issue is that it's not how to select just the best tool for the case. Is to figure out what are the characteristics of each dataset that have the biggest impact in the result that you get. One of the things that kind of struck me when I started working with somatic variant calling a few years ago was that there is no consensus. The thing that Stella showed with this big flower chart, I find it extremely, well, amazing that people rely on one pipeline only to figure out whether a particular somatic variant exists or not, especially in low frequencies. And this is kind of a bread and butter in precision medicine, which raises the question, it's not to figure out which is the best tool. I mean, at the benchmarking level, you can find this one out, but then it's not just the best tool, period. It's the best tool based on the data that you just provided, which data itself has its own characteristics. So it's a whole mapping exercise. Which data, what characteristics of the data, and for those particular pair of information, what is the best tool to approach for that? Does this make sense? Stella, something from your side?
Totally agree. There's no one size fits all, really. I hope there were, but no.
Oh yeah, that's an interesting argument. Yeah, so maybe that guys, Garmin and Ilya, do you have any other? Do you basically like see it the same as the because in germline again, as we've discussed, it's more there is like a better way or maybe not better way, but it's still more acceptable maybe to stick with a particular solution. It seems that it's not the case with somatic variants. So maybe from your experience, can you agree or disagree with this argument?
We also perform tests, and if you have Dragon, you have the answer. So it's basically performs better than all the tools that we did. We sequenced ultra deep tumors and then downsampled them, and then of course we were looking for the variants. And yeah, unfortunately, yeah, it costs money, this Dragon, but yeah, they're just better than everyone else. That's my opinion.
Okay.
I had experience with Mutect too, and FreeBIOS and routine practice in somatic colon variants, and we do test of benchmark all pipeline from wet probe dilution to colon mutation, and standard Illumina protocol can detect allele frequency about 1%, and if you you can up threshold to 5% if you want consecutive results in the standard protocols. If you want to determine to detect low frequency alleles, you should use special protocols such as UMI or duplex sequencing, for example. But it is a question, what mutation do you want to detect? For example, actionable mutations, which almost always connected to driver mutations, and then in the tumor, that mutations are in high frequency, and there is, for example, resistance mutations. In low frequency, for example, T790M mutation in very low frequency.
But in terms of tools, so that you said about the Mutect and FreeBIOS. So basically, what you if I got you right, yeah. So what you say is that if you're using the standard protocol and the frequency is relatively high, does it mean that both of the pipelines you've tested perform well for these cases, or?
Yes, FreeBIOS and Mutect can detect mutations in both cases. Mutect we used for paired data with norm and tumor.
Oh yeah, that's the important thing about the paired data. So as far as I can see from all of the opinions which have been mentioned, it seems that really there is a very big difference in terms of the actual frequency of the variant, because it seems that with the higher frequency variant, there are more chances that more tools will get it, and with the lower frequency variants, it seems that maybe there will be more difference. And as far as I understand, yeah, the benchmarking approach which have been presented in the first part, yeah, it seems that it actually will be easier to see which tool does better with different groups of variants. Okay, yeah. One more thing which I wanted to ask before moving to other aspects is sort of beyond the idea of looking beyond just variant colors. Yeah, because we've seen, yeah, thank you, thanks to Stella for showing this Venn diagram, which I think was nice to illustrate that there are many discordances between the tools. But again, the question is not only the variant color. There are other stages. There is the alignment stage. There is the pre-processing stage. And again, in germline case, we've mentioned in this on a previous seminar that in germline case we have lower dependence on the alignment. We have lower dependence on the pre-processing. And variant color basically plays the dominant role in terms of like defining the actual accuracy of variant calling. So what's for the somatic case, is that the same, or like what's your maybe perspective and your estimate of the relative importance of raw data pre-processing and alignment versus variant calling? So for Stella, if you could comment on like whether you've seen in your, for example, with the benchmarking approach, differences between not variant colors but different aligners and pre-processing like approaches. Have you tried it?
So, like I said, there are 4 main pillars in the variant calling process. That is, of course, the sequencing, the mapping, the variant calling, and then the filtering / annotation, and all the processing in between. So we have seen that the mapping and the preprocess, everything plays a role and affects the final variant calling process. And that's why I was very happy when I stumbled upon NEAT, because it produces the already aligned files. So I can start by a common starting point. So everything is aligned, and then I can take out this source of errors, and I can move on to the benchmarking. So the short answer is yes, everything plays a role in the final variant calling process.
Yeah, the thing, maybe for a short follow-up. So the question which I have is basically like the relative influence, because again, in the germline case, we've done this with a lot of like genome individual samples, the standard benchmarking samples, and the picture there is that, well, the variant colors, like the difference in the variant colors will be like in hundreds of variants generally, and the difference between aligners in many cases will be like tens of variants. So the differences between alignment will be like tenfold less influential compared to differences in variant calling methods. And so, like, do you have any maybe like personal impression as to what plays like a more important role with lower frequency variants? Because my understanding from a personal perspective is that for lower frequency variants, the alignment may be like relatively more important, because the placement of individual reads is more crucial than in germline case, where you have high frequency of alternative allele. So, like, is that true? Like, do you have any impression again as to the relative importance of this? So, is alignment, this affects basically the results the same way as variant colors do, or is variant colors still like the dominant factor here?
I can try to give a first answer, and then Stella can contribute as well. So the short answer is that the mapping itself does not have that much of an impact directly. I mean, the scale is the same, but the processing on the BAM file, which is realignment, repositioning, etc., these and sort of changing the flags again, these do have a significant impact, especially if you look at the false positives and false negatives. That's kind of the main issue that is then propagated down into the variant calling process, because most of the variant callers expect reads to be left aligned. And then when you start running the pipeline without this particular step, you start seeing kind of phantom variants emerging at low frequencies, which is essentially noise. But it's because, especially looking at amplicon-based somatic variant calling, which is panels and not whole exome, whole genomes, you start seeing that these kind of phantom random variants kind of emerge. So shortly put, it does have an impact, the mapper, the aligner itself, similar to the germline, but the steps between the mapper and the actual aligner calling do increase significantly. Again, I would argue tenfold the number of variants that that pop up along the way. So cumulative, those two ultimately have a significant impact compared to the germline. That's kind of my quick version. I don't know, Stella, do you want to add anything?
I think I'm covered.
Okay, thank you. Yeah, that's an interesting point. Yeah, about this intermediate pre-processing in between the alignment and calling. So maybe again, guys, German and Elia, did you have did you apply any sort of additional pre-processing? I understand that German, if you use Dragon, as far as I understand, the Dragon just produces the variant call, so you don't have any influence on what's in between, or do you?
That's difficult to say. Indeed, you can submit different types of tasks for Dragon. So probably you can just run the variant calling without the alignment itself. And I think from what I remember, what was done and kind of improved the performance is adding the HPV genome there and something else, I think, to the reference. So we have not only human reference, but also several viruses there. And this one improved at least in how informative the report is. Not talking about the calling of variants, of course, but just the other things that we want to know from tumors. Yeah, and that's more or less it from what I know.
So they mean that the decoy reference, so-called, yeah, which includes all of the okay
additional things.
Okay, yeah. So did you run any in terms of, as far as I understand, yeah, with mutex you probably use all of the GATK recommended pre-processing, like the realignment and base quality recalibration. Is that true?
We didn't realignment. Because mutex 2 don't need realignment. It realign inside reads. We actually cut adapters by BCL2FastQ. We put the sequence in a sample sheet, but if you do not do this, we didn't see any difference because adapters aligned as soft clipped bases, and you should only tune your caller to don't use soft clipped bases, and you can not trim adapters and another pre-processing. Only switch off soft clipped bases. It's all.
Can I ask a very quick question from my side, mostly to clarify? So far I've been discussing about somatic variant calling, but we haven't kind of disambiguated between the tumor only and tumor normal cases, which have both a significant sort of impact down the road. Dragon, to start with that one, is designed in particular to support tumor normal, and they have kind of an approach for tumor only, which is not 100% clear on my side, but I may be missing some points as well. They do, however, have a very significant component in the Dragon pipeline, which is that they rely on the external database, similar to 3,000 genomes, essentially what is a population kind of case of variants, germline variants, so that it can remove them, even if you're talking about tumor only. And the second point is because it's part of the Illumina framework, and hence it's very, very well tied to the Illumina sequencers, and they have a noise reduction module, which essentially removes any kind of expected noise. What in my mind is kind of unclear for Dragon, and that's kind of been emerging in our benchmarking efforts, is that if you're talking about variants that are low frequency enough that are equal to the noise, Dragon essentially scales things, everything out, and that's fine. Because in those cases, ultimately you don't care, or you can follow up and you can find out if you're kind of missing something at that point. On the other hand, if talking about MUTEC2, which does have a tumor-only case, the challenge that I see there, and that's kind of the question in both cases that I want to ask, is how you resolve the modeling, the internal model that kind of arbitrarily downscales allelic frequency, depth, etc. So if you input a system of like 5,000 depth in a particular case, you end up with a depth reported by MUTEC2 of like a few orders of magnitude below that. So I'm wondering how you've been approaching these kind of challenges in this case in somatic variant calling.
As far as I understand, yeah, that's the question to German about the relates to Dragon, yeah?
For Dragon and MUTEC2 respectively, yes.
I think we didn't use Mutek. I think we used Strelka before Dragon there. For tumor only is I think we work with tumor only with hematological malignancies. It makes sense. Otherwise, we work this standard clinical pipeline is tumor normal there. And when we work with hematological malignancy, you actually can expect that the purity of the tumor can be quite good, because you can use something like FACS sorting. You can sort the cells and sort out the if it's like T cell, B cell, like okay, most of the tumors I worked with, hematological malignancies, sorry, not tumors, were actually very good purity. And their clonality there is also not that big of issue. I would say like the smallest clones are like 10% that I work with, and it's already quite a lot there. So in our, I know that Germany is kind of let's call it a rich country, so the insurances can afford the sequencing of tumor normals there, and many other countries probably look for the ways around to sequence only tumor. But we, for the most of solid tumors, we use the tumor normal. So for us, it's not a problem there. That low allele frequency and no normal tissue for filtering, we don't have this. That's why it works for us. Maybe in other circumstances it wouldn't work. I hope I understood the question. Yeah. Okay. Thanks.
Yeah, that's a good, I think, a good point to talk about this tumor normal versus tumor only mode. And yeah, I understood the government's argument that basically, in cases like Germany, in most cases you do tumor normal and you do tumor only for hematological samples. Yeah, but yeah, indeed, and for other countries it might be a little bit different. But my question probably will be in terms of the reliability of the results. Yeah, my impression, which is not motivated in any experience but because I virtually don't have any experience with that. But my general understanding is that, well, I expect the tumor-only results to be like much more noisy, yeah, and require much more manual maybe curation and heavy filtering. So is that true, or with the modern approaches for variant calling, is that true that tumor-only calling will be much noisier than tumor normal pair, or not? Maybe Faris, if you can.
Yeah, I can answer that easily. So yes, the tumor only by itself is inherently more noisy, mostly because having the tumor normal pair, a lot of the variants that have been identified as germline are removed. Plus, it can allow for a modeling of the noise, because if you see in the germline a particular pattern, you can use this as a mechanism to remove noise from the tumor as well. That being said, even the tumor normal pair doesn't really solve the issue of lower frequencies, which is the main issue. So in hematological malignancies, as was very well indicated, you can have high quality data, but at the same time, a small frequency can ultimately lead to a very huge peak of the disease in a few years, which makes it more imperative to make sure that these low frequency things are captured early on. In solid tumors, this is not as evident as far as I know, but I'm far from a medical doctor or even a biologist, so you can easily ignore me if I'm talking nonsense here. But in both cases, you need to have a robust mechanism of removing noise. Both striatal and dragon have their own kind of noise system embedded in the process itself due to Illumina. But if you try to use this tool with non-Illumina based sequencing, then it comes, well, either you cannot run it, or even if you manage to install it, you cannot really use all this denoising process. And again, if you go back to your 2, even if you have tumor normal or tumor only, in both cases, there are a few challenges resolving to the reporting of the actual frequency and the actual depth, which is the other major challenge when we are doing benchmarking.
Okay, yeah. So, German, with regard to the samples that the tumor normal to tumor only cases which you've had, and maybe you can compare it to the, as far as I understand, yeah, you've mentioned the sorting of cells for hematological tumors. So the first question will naturally be, do you have experience with like sorted and unsorted, and so what difference does it make? I assume that it will a huge difference. Yeah, and then like what's your impression about working with the results for these and paired samples? Is that really like much noisier in there?
Sorting or no sorting, I have no idea, so I cannot claim any knowledge.
So sorry for interrupting. Yeah, so I mentioned German because German mentioned explicitly that he worked with sorted versus with sorted tumors. Yes, that's what motivated my question.
Yeah, sure. With sorted, you may expect the biggest clone to be close to 100%. Makes sense. There and you care only about the driver mutations, and many of these driver mutations are, from what I know, they are also causing the rare diseases, for example. Yeah, it's a famous example of these genes which are responsible in germline for neurodevelopmental disease, and in somatic they're responsible for the cancer driverness, let's call it like this, like DNMT3A, this gene, or SXL1. So when you interpret these variants, you can when you try to figure out which actual driver mutations, you can kind of look what sense this variance, because indeed the allele frequency of the sorted malignancies will be close to 50%. For heterozygous variant, it will be close to 50%. But then you may try to understand what these variants mean. Is it realistic that this variant is in germline? And what would it make sense for the tumor? If it's somatic, is it driver mutation or not? Yeah, some genes don't produce. There's very important for the hematological cancer, like TET2. It's an important cancer driver, but it doesn't produce any phenotype in germline. So here you may be, you might ask yourself a question: is it germline variant or is it somatic, and then it drives the cancer? But otherwise, I think it just like it takes time for the interpreter. If you really need to pinpoint 5 or 10 important variants, it just takes more time for them to interpret them. But in the end, it's possible to roughly still say, is it likely germline or likely somatic there in this case.
Yeah, even in the absence of the normal control, yeah? You mean?
I would, yes.
Mm-hmm. Yeah, that's interesting. Yeah. So, and Ilya, did you have I assume that you mentioned that you've worked with paired samples, yeah? Have you worked with tumor only?
Yes, sure. We worked with both. Because the partner clinics, when they work with the blood malignancies, they provide only tumor only samples, and then that's why I had. Yeah, I already said it in the previous seminar. I developed my own tool for the variant calling, which is CNV calling, not short variant calling. And then, of course, we had to invent these tumor-only modes there. And yeah, indeed, it produces quite a lot of issues there. And for some of tumors, it's absolutely impossible to call. Like for the complex tumors with the polyploidies, it's just like something that I would recommend against, because it may hurt the patient in the end. Because you will not see the polyploidy in tumor-only most of the time. Like at least I wouldn't see. Maybe someone knows some methods. Yeah, so it's recommended to work even when you do this FACS sorting thing. You can also sort the normal cells separately, and you can sequence them separately, and you may still sequence tumor and normal. So it would be preferable way. And if you for some tumors, I guess it's really possible to work with tumor only, which are like not a lot of clones, not a lot of drivers, and the structural variants are not that frequent there, which is like the CLL, chronic lymphocytic leukemia, for example. It's not very destroyed, the cells there, the DNAs, there are like, I don't know, 5 CNVs and 5 point mutations overall. But for the early tumors which are significantly hurt by the mutational process, I wouldn't recommend doing tumor only at all. I think it's just like against the practices. It should be prohibited, let's call it like this.
With regard to tumor only, yeah, thank you for the comments. I've seen, yeah, and here I will mention the mosaic variants, which are essentially like they are naturally somatic, but you do the analysis of mosaic variants, I think, for the diagnostic purposes, in like when you not working with tumors, for example. I know that you're just diagnosing rare disease. And I have seen though that with mosaic variant callers, many people like there are best practices in their papers which recommend doing some weird but still quite I think effective strategies, like using germline calling, like GADK, but instead of like using it in the default mode, using it with a ploidy parameter like 50. So assuming that that's like a 50 ploid cell, and then doing it in a germline way, and then like using these as variant calls. So my question here will be, in case when we're working with a tumor-only data, like is that like a like valid strategy to use something like the callers which are not specifically designed for tumors, for somatic variants, like plain GADK, like unified genotype or haplotype call, but configured for with these specific like ploidy regimes, or is it only valid for mosaic variants? So is there like any, because that's actually what I didn't have any experience with working with tumor normal pairs, but we've had a couple of hematological tumors like 5 years ago probably, which we've also tried to call like with plain GADK haplotype caller with some modifications. So maybe, so again, the question is whether it's a valid strategy for a tumor-only mode, or should we stick with the somatic variant caller specifically designed, even if it's a case like hematological tumor.
I can start, and I fully agree with what Jermaine was saying. Like, no, this is not a good idea. You can get results, but I will definitely not rely or believe results from, let's say, 3 Bayes on tumor-only samples by setting ploidy to, I don't know, 15. I'll give you low-frequency variants. Probably some of them will be, well, not true, but some of them will align and be the same as the one that you would be getting in another approach. But ultimately, you're forcing a variant caller designed with a completely different stochastic model to give you results on a process that does not fully align with this model itself. So to get results, technically, I would not trust them. That's kind of my quick and dirty version.
Yeah, that's thank you. That's an important comment. Yeah, because that was my impression as well. That probably it's not a good idea. But the question is then again, German, maybe have you had the experience of like mosaic variant analysis in rare disease?
Yeah, sure. So, but
What did you use, like the somatic variant covers for this case, or germline, the specific configuration?
I don't think that this 50 cell thing, 50 ploid is a good idea also. It's like something like if you have GATK set up on your cluster and nothing else, then it may be okay. But overall, like these prior probabilities, why would you do this like 50? If you do have ultra deep sequence sample, you will still miss this variance because your prior model of this 50 ploid cell, it will reject the variant because it will be implausible there. So it's something I would recommend the cases thing. Just use proper colors. If you don't want proper colors, there is VARScan tool, which is like you just set up like 5 grids and you call everything which has 5 grid support. You call it, and after that you may filter it with like this strand bias, something like this. But I think this JTK idea is just because you have nothing else on your server. Why else would you do this? And yeah, for mosaic variants, there is also the specific wet lab. There is ultra deep sequencing, of course. If you suspect mosaicism, we'll probably go into this direction. And also, yeah, it's important from where you take the tissue. So there is whole specific. It will be enough to talk a whole seminar about mosaic variants in rare disease. So it's specific.
Okay, but the answer is that you don't use like these like traditional like somatic pipeline, the same for as for you do for the tumor only. Like, you don't use the same for mosaic variants. You go for the specific methods. Yeah, that's the answer.
I don't see why would you use methods with the assumption on the ploidy in case of mosaic variants, because you really have no idea. Yeah.
Yeah, that's yeah. I'm citing it just because I think that was a genome biology paper, like which is called best practices for mosaic variant. Well, they were specifically they were talking about whole genome sequencing with 250 plus coverage. So that's probably the answer, that they have like they really have a whole huge depth. But still, yeah, that's that's interesting. Okay, yeah, but about mosaic variant course is indeed not the topic for today. That's just important to mention, yeah, that it requires a different set of methods.
Sorry, just short interrupt. Huge depth in somatic variant code is 10,000 or more. 250 is not a huge depth. 250 is just like math.
Okay, yeah, and that's it. By the way, it depend on whether you do the tumor only or tumor normal setup. So what are the coverages that you can expect in both cases? Yeah, does the tumor only case require higher coverage generally?
Not necessarily, I wouldn't say so. If you again, if you work with the very pure and very homogeneous tumor, you don't need very high coverage there. And yeah, it really depends. Like if you sequence panel of cancer drivers, which is like 600 genes, then you allow much higher coverage. If you sequence whole genome, because you look for the non-coding variants, or like you're sure that the fusions are very important in your tumor, then you may go with shallow whole genome sequencing, something like this. So you don't even need to sequence that. Yeah, deep. It depends. The approach depends on the typical. Different cancers have different drivers, different signatures there, and based on this, you may. If you work with the patient with the first stage tumor, you may select one approach, and the treated tumors of the fourth stage with like 3 treatments and all of them failed, then you would select a very different approach, in my opinion.
Okay, thank you. Thank you. Yeah, as we I think already mentioned the filtering issue. I think that we can move to talking about filtering and like selection of the actual variants we're looking for in the data set. So when we were discussing germline, again, I will refer to the previous meetings. Again, in the germline case, we have been talking quite a lot about the strategy, and I think came to the conclusion that it is better to have fewer filtering upstream and allow more like stratification of the data by the people who are interpreting. Keeping more of the potential artifacts at least just so that the people who are interpreting the data are able to access them and maybe go one by one in case they don't find anything in the confident candidates. But I assume that, yeah, when we're talking about low frequency variant, etc., there is the picture should be a whole lot different. Because yeah, we'll get probably much more noise, much more variants which are detected. Yeah, with very few reads, and in this case, I assume, again, this is my assumption as a person who is not very experienced with somatic calling, that there will be more more necessity of like specific setting specific sets of filters, and yeah, more rigorously selecting the candidates, because otherwise, yeah, I assume that the space for like looking into like the candidates that you can consider will be too large. But again, this is my assumption, and so my question is, in terms of filtering the variants, like what is the strategy which you see as the best one for identification of let's say like driver mutations or any other kinds of important variants you can be looking for in a tumor? Again, I'm not a tumor expert, so I'm not that's experienced with like what particular kinds of mutations you may be looking for in the data. So again, the issue is should the filtering be like more stringent or more relaxed, and what features of the variant will you be using primarily for filtering of candidates?
Well, that's kind of a question that I honestly have no idea how to respond to, because it largely depends on what you want to achieve at the end. German mentioned that if you're doing like deep sequencing, you have like 9,000 depth or more. At that point, having a filter that sort of takes out everything that is below 10 is kind of common sense. On the other hand, if your coverage is around 500 and you remove everything below 100, then you're really missing like 20% of your stuff there. So that's not a good threshold. So what I'm arguing is that this is, as far as I know at least, or from our side here, there is not one consistent strategy that you're using homogeneously across kind of any sample. We have kind of a different setup for whole genome, whole exome, or amplicon based. And again, depending on the actual wet lab experiment, how well it went, what are expectations, and more importantly, at the very end, as you very well said, most of the things are going to be reviewed by the experts together, possibly with IGV open on the side, to review each and every one of the final called variants to see whether they actually do exist or not. Because another kind of fun stuff that's happening, and thankfully this is not something that it's not that we're doing it, but we're trying to resolve it, is that if you run two pipelines, even with all the stringent filters and everything else, the ones that are called with very high confidence, using, for example, one hand Strelka, which we also have here, and the other hand MUTEC2, you see that there might be a variant that is called by MUTEC2 and it exists, but it's not called by Strelka, and vice versa, which means that ultimately, after all the filters, you have to go back to IGV, and the expert needs to look at each and every one of them and see whether they actually do exist. That's kind of the pain in the pain that our sort of colleagues have asked us to resolve one way or another, and hence Stella's whole PhD.
Okay, Garen, do you have something to add to this? Yeah. Okay, yeah. And so basically, yeah, maybe Ilya, do you have any maybe other perspective on this issue?
We annotate variants by databases such as Cosmic for actionable mutations, which related to dedicated therapy for each gene, for example. And the next for mutation, which absent in databases, we prioritized variants by CADD score. CADD score is relevant metric for somatic mutations too. As we try compare CDD scores in actionable mutations, known actionable mutations, and they have high score in CDD.
So what I'm talking about, yeah, thank you. That's about the functional prioritization, yeah. And did you consider like any sort of additional technical factors, and or do you just like take all of the calls and then just annotate them and look for candidates? Is there?
Personally, what I know that we don't work with tumors or subclones less than 20% of the standard. We use, I don't know, approximately 300x for normal and 700x for tumor, the coverage. And if it's less than 20% of purity or the clone is smaller, we reject all these variants more or less, because yeah, if the tumor comes like this, it's irresponsible to give any recommendations based on the material. So we ask pathologists to provide us better tumor sample there. And yeah, that's more or less, it's very industry-like approach, not much science here, as I see, but yeah, in the clinic, I think we have to put some quality limitations there.
Yeah, so that basically means that the quality of the material matters a lot. Yeah, but when you have the calls, you don't do as far as I understand it, what all you have said. Yeah, that you don't do specific filtering, additional filtering based on technical parameters, but rather you do functional annotations on the one hand and visual inspection on the other hand, and then trying to look at the candidates which are suitable using the functional metrics and reviewing them manually with IGV and other tools. Yeah, does that seem like a reasonable strategy? Yes, you're taking the calls, not really looking much into the specific technical parameters, but then doing the functional annotation and then proceeding to review. Is that okay?
When I said 20% tumor purity, it also puts the limitation on the technical parameter. So you don't work with the variant allele frequency less than 10%, I think.
Okay.
In the end, it's 10%. So you perform the filtering of the variants, which maybe not 10%, maybe 5% for 20% of the tumor content. We may expect up to 5% maybe going. So these ones we all filter out. We consider them unreliable there. And yeah, if you don't have enough drivers there, there is nothing to discuss. It has to be realized at all. So we put these hard thresholds, the hard quality things, and then nowadays it's not only the driver mutations. Now also people look for example, signatures there, and for these ones, the technical requirements for the quality for these mutations are a bit lower than for the therapeutically important, for example, mutations or cancer drivers, this one. So I would say even within somatic calling, there are several processes in the end. And for one process, like tumor signatures, you may allow lower quality variants. And for another process, you have to put different quality filter there.
Okay, thank you. Yeah, for this, maybe if you can please share what's your maybe not the only this question, but just generally outline the pipeline of downstream processing. So if you have a variant call, so let's assume that you do have a sort of let's say trustworthy calls in there. So what's the downstream pipeline is working with this data step by step in terms of annotation and then review. If you can outline the whole procedure of what you typically do after you have the calls.
Well, this is the point where it goes beyond our hands mostly. So I don't think I'll have more to discuss on that. Mostly because, as Ilya said, after you do filtering, you have to do annotation. And there are a million different databases that you can do annotation for. And the selection comes down to the problem. So depending on the group that has access to the variant calling, we have a different set of database that we can use. There are some tools like ANOVAR that allow us to interrogate multiple databases at the same time. There are custom-made tools that allow us to interrogate particular databases that are not API accessible, but you need to have kind of an intermediate step to get information out of it. And you also have the in-house databases, like the one that Illumina has for noise. Similar ones exist that kind of capture variants that have been annotated custom by the end users, the experts, or medical doctors or biologists, and then we can use this as annotation. But ultimately, this is not a very complicated step, because as soon as you have the variants, making the annotation themselves is mostly a process of figuring out what is the appropriate database, or appropriate, that's not the correct question, the relevant database for the challenge at hand. And then you provide this information to a easy, consumable way to the end users. People generally don't like VCF files or similar, but they do tend to love CSV files and Excel files. So ultimately, at the very end, this is what you present. Occasionally, we've ended up creating kind of interfaces to view those more interactively, but I cannot claim that these have been a huge success, mostly because maintaining them and sort of keeping up to date and providing all the functionality that pops up along the way will be almost equivalent of like recreating the whole Excel system. So might as well use Excel from the get-go. So that's kind of the quick version.
Okay, thank you. Yeah, regarding the interpretation part, yeah, one more thing which I am personally interested in, again, is for germline variants. I understand that there are strict criteria. You have a CMG, which provides you a very good framework, which like essentially is in many aspects like simplifying the process of making some final decisions. And that's, again, this as far as I understand, yeah, for the somatic case, it might be a little bit more tricky. So my question is, like, are there any criteria? Maybe they are specific to a particular, like, to particular domain, to particular tumor types. Are there any criteria which are like formal, not dependent on, let's say, the discretion of a particular interpreter, but which are codified the same way as a SEMG for interpreting, like selecting like driver mutations for cancer? Are there any things like that?
I can try to answer this. In my opinion, there is nothing like a CMG there, but what there is typically about any variant, we can say what kind of level of evidence we have against this variant. This variant actually plays this role that we think. It may be the level of the early studies, it may be a level of large cohort study, it may be a level of the preclinical study, a level of already finished clinical study, which is the highest level of evidence. So I would say each variant is it colored in different color according to the level of evidence that this variant actually plays this role in the tumor. I would say this.
Okay, but that's still that makes the so there is the evidence level, and you don't, if there is no specific like functional evidence, then what do you do with that? So basically, you just consider that a plausible candidate, but so what is supposed to do? So is it that, does it like have to go to some functional validation or what? If you have, let's say you have a particular gene. Yeah, and you have a variant. So with this evidence level, maybe I misunderstood you. So is this evidence level specific for a gene or to a variant?
Both. There are many recurrent variants in tumors, of course, and many drivers we can see in many different tumors. And typically, we don't have a time to do functional validation. If there is a patient with cancer, there is really no time to do cell culture to test if it has cancerogenic. But what there are many these experiments of massive parallel assays of mutations. So they were like introducing like a rain of mutations in different cancer drivers. And from there, roughly, we can estimate the fitness of the cell. And there, they were able to. So it's one of the techniques that they now try to apply to rare disease, but I guess less successfully. But for cancer, because it promotes the growth of the cells, it kind of roughly gives the idea if the mutation has any functional effect or not. So there are different sources of evidence there. And I guess functional validation is for many of the patients is not an option. It's just like there is no time for this. If it's unique variant, not seen anywhere, and you doubt that it makes this like protein truncating variant or the frame shift in the beginning of the gene, it's quite clear it turns off the your gene. But what is like missense variant in some you haven't seen any variants in this particular amino acid there? What can you do? You can look at this massive parallel assays. That's the only thing you can then do. But giving the clinical recommendation based on this, I'm not sure. It has to be this. There's a special board, tumor molecular board in our clinic, which does decision based on this, and I doubt that they make a decision. But I cannot provide any details because I'm not there.
Okay. Yeah, that's interesting. Yeah, thank you. Yeah, that's I think all with my questions for today. I just have only one thing which I'm personally interested in, which is you've mentioned German mentioned and I think Fotis as well, the concept of tumor purity. So my question is, like, what is the estimate of that? So does it come from a wet lab or is it done like computationally? So what's the
I think that's, well, as far as I'm concerned, this comes from the wet lab directly, from the people who are working with. I don't think that I can assess purity of the sample just from the computational process. Indications might emerge, like you can have low quality information or you can have a lot of noise, but I don't consider this like a definitive indication of like whether you have a pure sample or not. So that's for the wet lab from my side.
We use both. We use computational prediction but computation prediction is not done based on short variants. Computation prediction is done based on the large variants. If there are no common number alterations there, you cannot really. Short variants are not very reliable unless there are tons of them, and then you will put these density plots and try to decompose your tumor. But if it's tumor only, then you will have issues with this. But when it's tumor normal, then you use computational many CNVs. In this case, you can estimate and usually they're concordant, but of course it happens that pathologists were estimating one piece of tumor, and for sequencing another piece of tumor was done. It happens regularly, and sometimes we totally disagree on the estimations. But we were able to show that the good correlation. Not only us, many people who developed the CNV colors for somatic, issue. They showed that the concordance is good, and we also did this. So I would say our doctors, they mainly rely on the computational prediction in the end, because pathologists also has quite a lot of. They need to count the cells. They need to look at the microscope and count the cells, and it's not an easy process. Hopefully, artificial intelligence will say something in this area that they will count cells more efficiently. But yeah, currently, I would say this DNA-based prediction of purity is what they use. But everything is broken when the tumor is polyploid. Then it's like it becomes troublesome.
I see. Yeah, that's interesting. Yeah, that the computational is also possible. Yeah. Great. Yeah, thank you so much. Yeah, that would be all questions from my part. Yeah, that was like very interesting and useful for me at least. Yeah, thanks you all for participating. If there's anything you'd like to add or ask, like then please feel free to do it. And if not, then we can conclude today's meeting.
Thank you very much for organizing it. You have been an excellent moderator.
Yeah, thank you. Thank you for joining us. Yeah, it was a pleasure. Yeah.
Bye, everyone.
Hope
Thank
to
you.
see you next time. Yes. Thank you. Bye.